let's see if you can get your head around. So this is similar but different. So let's go over, I think, what you should know. So what's the name? And we can just do this together, okay? To kind of speed this up today because we're a little shorter class. So you should know, what's this? And here? What's this? Can you see what this is? What about this one? Are you sure? Okay. What do we have here? So what will this become? Primer. You guys are on fire. Oh, what do we have here? So is it on or is it off? Off. And so what would turn it on? They're not always as good. Usually they're throwing things at me. No, they're not. So, okay, you got all that. So that's the exact model that I taught you wonderful people with, right? So look at what I built here. So is the meter stick on parent or daughter? Parent. And here? Parent. Okay. What would we call this part of the bubble? Origin. So those primers, okay, now I'm going to go individually here. These primers here at the replication origin, what strand, and we have two, right? One, they're moving in opposite directions. Two, what do we call those strands that were produced from those? The leading strand, okay. What do we call these strands? And why did I stop DNA polymerase 3 here, do you think? A silence, yeah?
Because it hasn't finished that chain yet.
Correct, correct. So what would we call these two sections here up here? Just the DNA. So what's the name of this enzyme here?
DNA polymerase 1.
So what's DNA polymerase 1? Is it going to connect Okazaki fragments?
Yeah.
How does it do that? Yeah.
It replaces the primers with their corresponding DNA.
Okay, good. And you notice that I just put one RNA primer here to save space. What's the purpose of these SSBs?
Keep it
Christina?
to keep it from tangling up.
Okay, and what's it? The parent strand. Okay, the leading or the lagging? Okay, good. Okay, well that was pretty solid. So that's our memory of DNA replication. So today, who can tell me, what is this a memory of here? So if I do this, right, so I happen to pick out pair 18, right? So we're going to follow one pair of chromatin from the genome in a lesson that's going to lead, if we have enough time, if not we'll start tomorrow, but it's going to lead into cancer, right? So I'm just going to take this pair, and you're going to start making a note. I'm going back up to the board.
You put it back there.
We gotta move all
Should we
this
move
stuff.
everything?
Yeah, that's what I'm saying.
No, we have two primers
Here,
on
this is what we're gonna do, buddy.
Yeah,
We're gonna pile up all the oxygen.
Wait and see it.
We need to move everything though. It's kind of.
Are you need to clean everything up?
Yeah.
Just put it up here. Move it
What
up.
I'm doing.
Just push everything down.
I'm putting all the carbons over here.
Put it here. Put it here.
Now we're
There
doing
we go.
There.
They all just connected together. 11, 14.
Let's start with 2. Do we have anything? Like, do we have?
Do you have the primers?
We're supposed to get.
DJ, they're right here. I'm clearing space right for you.
Danny gave me too much of each.
Yeah.
Hey.
Okay, here we go. Move this out of the way.
open like,
You got the note?
yeah.
Good man.
Yeah,
Here, I'm gonna
open it up more.
So we could add 11's at the top, 14's at the bottom.
A primer. Something like that, right? Like that.
Do you have
What?
do you have the primers?
That's the. Close enough.
polymers.
That's like the mini bubble. Right?
Oh, it's okay.
Yeah. Okay. And then initiate. I mean,
Yeah, because I can
I'm just waiting. I'm just waiting. You just destroyed the station.
alright, well there's only A's and only G's, barely C's.
Yeah.
I got 2 G's and 2 A's. So I should have 2 T's.
You have DP3? Yeah.
No, what? I don't have anything. I just got these. Just take those. Yeah, it doesn't matter anyway. I don't think it doesn't really matter. Actually,
I think we.
no, wait. Oh, so these would be the primers. So, yeah. And, or which is, yeah.
Can I model it at my station? What am I mixed up at? Why am I mixed up? That's all practice, right? That's exactly what that is. So, speaking of practice, on Friday I built this for you before I left. So this is going to be our magnetic memory of what lesson? DNA replication. What's the name of this protein? What do we got here? Yeah, notice how I put it on the top? Like, I tried to really emphasize that. What do we think here? What do we got here? Can somebody tell me one significant control mechanism that P53 has? Yes. Okay, good. Can you get a bit more specific? Like, I like the language. Donovan?
It fixes mutation.
Pardon?
It fixes
It
mutation.
fixes mutations. What does P53 do, Hunter, from a regulation mechanism if there's just way too many mutations? What can P53 signal? You got that? Sends an apoptotic message. And in simple language, what's an apoptotic signal?
You're just told to stop replicating.
And
It'll just kill it.
it's a little aggressive, but it's correct, right? That the cell will go through natural cell death. Okay, I shouldn't say it, I can't help myself, but like the leaves. I apologize.
I'm right here, man.
No, you know what, as I tell my friends back home, it's impossible to be a hockey fan and live this close to Toronto and not become a fan. So I was hurt last night too. I'm just excited about what Pittsburgh's gonna do.
Oh, yeah. Oh, I hope they crush them.
Yeah, I think, okay, let's go back here. What do we got here? What do we got here? What's this going to become? What is the name of these 2 primers at the beginning of our mini bubble? No, right where the bubble starts, that's where helicase and primase enter. You're one step ahead. Yeah, that's the replication origin. Okay? Okay, two other things we'll get to the lesson. Is the meter stick on leading or lagging? Okay, is the meter stick on Okazaki or leading? If I said Okazaki and lagging, both. What's the name of the enzyme that's gonna remove the primers and replace them with? What's the name of the bond that ligase is gonna make? Right? Covalent bond. Okay, beautiful. Okay, let's go. So what does that magnetic memory represent?
The human genome.
The genome. So how many pairs of chromatin are in that genome? 23 pairs. How many am I holding? How many pairs? Okay. So you see on the back, we did this before, pick pair number 18. Okay. So you're going to start by making a fairly straightforward note, then we're going to figure a bunch of stuff out here today. So this is a simple picture of the human genome. Pick up one pair. So you build that picture and off we go. Okay, rock and roll. We're going to start at the end for our guest today. So I guess you could call this note the cell cycle. There's a bunch of stuff that's a review, and then there's a bunch of new stuff. Okay. Let's just deal with what I think some of you have been taught. If you haven't been taught this, no big deal, you're gonna take a note on it now. Okay, so what's a telomere? Where, any idea what a telomere is, where it is? Have you heard this before? 1, 2, 3, 4. What else I teach in here? 5, 6. Can't remember the old telomere? Anybody? Yeah?
It's like a biological clock.
Okay, good. It is a biological clock for sure. So a telomere is a sequence like this, right? I'm highlighting it with monster molecule. Right, so it is this tag, it's easy to remember, TAG. TAG. And it's on both ends of the chromatin. So, let's grab one of my shoes. Okay. So Donovan, you need to get this down, buddy. Okay, what am I touching here on this shoelace? How would you describe what I'm touching? Am I touching the lace? I'm touching the lace right now, but now I'm not touching the lace. What's the material that I'm touching? Plastic. Is this plastic cap in the middle of the shoelace? Is it a pain if that plastic cap breaks? What will happen if this plastic, or when these plastic caps break? The lace starts to what? Break. Good. People are sharp. So one of the things, if you look at it from this point of view, right? So genes, we haven't started this yet, okay? Remember, chromatin number 22. That's the shortest chromatin in your cells. How many nucleotides are in chromatin 22? Big guy?
50 billion.
Attaboy, 50 million. That's more than the population of Canada, right? So, we haven't begun this component of the course yet, but we're going to quite shortly. Telomeres are not genes. They're non-genetic. so they don't code. Do you have any idea what a gene, okay, we have a coder in the room, right? He's going to do some amazing work I'm quite excited about. But he's going to write code. What's the code? These genes are a code for what? What are they a code for?
Protein.
They're a code for protein. And again, we're going to study all that stuff in detail. So the tips of your chromatin are non-genetic. It doesn't mean, right, that's what's so cool about this story. Thymine is thymine, right? Adenine is adenine. Cytosine is cytosine, but in this location, it is not interpreted as a code. It's interpreted to protect. Now, because of that, what is that strand called that the Okazaki fragments are on? The what strand? So, because of that lagging strand, I won't get into the technical component of this. Look, each cell division, all chromatin lose a chunk of telomeres. So you want to jot that down. So we're losing these things called telomeres every time a cell replicates. Now, let's freak out for a minute. Let's go old school. Never underestimate the power of shock, okay? So look, I don't know where you are there, buddy. Okay, so look, there's you and me, one cell, conception, okay? That cell, what did it do? Divide it, what did that cell do? Divide it, what did that cell do? Divide it, etc. Okay? Now, that's cell division. As one of my former biology teachers, who was an amazing teacher with it, I'm stealing this cool idea right from his brain. That cell division continued to make you and me. But what's profound about this is, if you take a line here, this is conception, this is birth. 9 months, right? 9 months. You as an organism, as a healthy person, live a long, healthy life. Let's say everything's cool, you die a nice, natural, easy death. Brady, where are you? Not violent. And you live for 90 years, okay? So you lived after birth for 90 years. There's more cell division from conception to birth than there is from birth to 90. Now, some of you, have you been taught by Miss Quinlan? So those of you who've been taught by Miss Quinlan knows that she just has come back from maternity leave, her second beautiful child. And as I talked to Miss Quinlan, children that are born, not even born, before they're born, up to age 2, just try to get your head around what I'm saying here in terms of cell division. They are producing 1.8 million new synapses per second up to age 2. So a synapse is a space between brain cells. 1.8 million per second for 2 years. So after birth, that's the rate, and there's a lot of cell division going on there too. I'll repeat this. There's more cell division from conception to birth than there is from birth to age 90. Okay? If this loss of telomeres continued, which unfortunately it does in a very, very small percentage of people. When I say small percentage of people, I think there's 9 people on the planet Earth right now who have this problem. They were born old because they lost telomeres during this first 9 months, And see what I'm doing here? The telomeres did not get put back. Now there's this crazy cool enzyme that puts telomeres back. What's it called? Telomerase, right? I happen to have it built here for you. Where is it? There it is. So what I'm trying to get you to think about here, this is a, okay, so make a little picture here of this amazing enzyme called telomerase, okay? You have it, I have it, but we don't have it everywhere, and there's a reason for that. Okay, I'm going to move this over here during embryology. So, the little baba's growing like crazy, right? Unbelievable. Every time that super mini human cells are dividing, which are crazy fast, all the chromatin are losing telomeres. But the defense mechanism is this enzyme called telomerase. Telomerase can find, right, the telomeres like this, right? And then telomerase will reattach, boom. So you build a picture here of telomerase reattaching. I guess I'll go this way if I'm following my pattern here, I apologize. So, a simple picture of the enzyme telomerase delivering a lost telomere back to the chromatin. And now we're going to try to get into some crazy cool biology of what's going on, why is it going? Yes?
If that just reattaches it, how come it falls off in the first place? Is it the same way
Well,
it gets
that's
reattached?
an excellent question. I'm not teaching you the mechanism how it falls off, but it has everything to do with the lagging strand. Visualize the primer. Can you do this in your brain? Where that last primer's put. DNA polymerase III bonds to it. Oh, because of the way primase is putting that primer down, it doesn't always put it right at the end of that lagging strand. There's some DNA that, so the primer, let me put it to you this way, the primer, here, the primer, here. So this is the end here, right? The primer should be put here, but the primer's put here. And that's where DNA polymerase III is going to start reading. So there's always a section in simple language. Okay, now, let's jot this down. So telomerase reattaches telomeres. That's why grade 12, I'll use a number that maybe you'll be comfortable with. I don't want you to freak out. Let's say you decide to have your first child when you're 30 years old, okay? You are not going to pass on chromatin that are 30 years old to your children. Why? Because right now in your body, you can jot this down, your sex cells have telomerase working. So at conception, the child starts off with monster long telomeres. The telomeres don't get short. Now this is where genetics gets interesting, and see if you can follow me here. If I'm not making sense, make me make sense. Telomerase, see my crazy line here? Look, 0 to 9 months, telomerase works in every cell in the child, the baby, the fetus, the embryo. Telomerase is working. Those children that are born old, that disease is called progeria. They do not have telomerase working. So that's why as they develop as an embryo, as a fetus, they're aging rapidly.
So
Okay.
telomeres are just falling right off.
Correct, yeah, and not getting replaced. So here's what's interesting though, because now this has a direct connection to cancer. So I want you to make a little triangle here like this. Okay, can you do this, little triangle? Like this. So, we have, what are we going to put here? Cell division, right? And here, you're going to put getting old. Why do we age? And over here, we have cancer. So cells dividing have everything to do with why we get old, and cells dividing have everything to do with what cancer is. And it's interesting that the people who study why we get older are teaming up with people who study cancer, because they're actually studying the same cellular mechanisms. So let's deal with this aging process. I'll simplify it to this picture here. The genes are protected by the what? Telomeres. Is genetic information the code for proteins? Is this important?
Yeah.
Okay, so the cells have this very cool brake, like there's a brake on your car, and the brake is the length of the telomere. Because look, if this telomere gets lost, and this telomere gets lost, what's going to get lost in the next cell division? A gene. The cell will not allow that to happen. So, you want to jot down that when telomeres get really short, okay? The break in biology, that break is called senescence. It's a fancy, cool word for cells will no longer divide. Because again, the cell is committed to protecting the most important component, which is the genetic information. Too short, yeah. So when the telomeres get to a certain shortness, that cell is done dividing. Okay. Now, I'm going to try to teach you something here. I don't know if it's how much sense it's going to make, but I'm going to try. Let's say this is chromatin 1, okay? We could look this up, but I forgot. Okay, let's say on chromatin 1. Can you do this, superstars? Look. The gene for telomerase is on chromatin 1. So there's a code on chromatin 1. I don't know why that, oh, that's normal time. Continue. Sure, sir. Okay, there's a gene on chromatin 1 called telomerase. So you gotta get your heads around. You already told me. What did genes code for? Protein. So you saw you saw the reality in one way of the gene. Genes code for proteins. So, can you figure this out? Let's go back to my crazy diagram. In the, yes,
What's the gene on chromatin knockout?
telomerase. So there's, as I mentioned to you before, there's a lot of genes, grade 12s, and a lot of proteins that have the exact same name. So is there a gene for DNA polymerase 3? Is that its name? Yes, right? Hexokinase protein, hexokinase gene. But here's what I want you to think about. Is the gene for telomerase on every cell during embryo development? Does every cell have the gene turned on? If the gene is on, you make the protein. Okay, jot that down. So the gene is on. So here's a world we're going to walk into, okay? Look at what you know already. Let's do a quick review here. Okay, so look. Right? Okay, we've done this. You should be able to figure out. Okay, so look, if we're watching this enzyme function, you've seen this many times, I think, I hope. Boom, glucose is getting phosphorylated. What's the name of this enzyme? Hexokinase. If you see this, right? What's glucose 6-phosphate bonding to? An allosteric site. What's that going to do to the shape of hexokinase? Well, close it, shut it off or on. So what is that word we've used a lot of? That is a what mechanism? It's a regulation mechanism, right? Control. So think of it like this. This gene is on, right? The baby is born with normal length telomeres. Any idea of what would turn the gene off? Two interesting things. What would turn the gene off, and why would this gene, that literally biologists called when they discovered it the fountain of youth, why would it be turned off to begin with? But let's deal with the first mechanism. How would you turn this gene off? Any idea? Okay, that's a tough one. So just here. Proteins bond to the genes to turn them on and to turn them off, okay? So just use that knowledge of hexokinase and that allosteric regulation. Molecules are molecular switches, on-off, right? You're going to be doing on-off switches. This gentleman over here was telling me too, sorry to get sidetracked, but he's in the process of building artificial intelligence, right? Just going to build the human brain. Why? Because he can. So he's dealing with electrical switches, right? Zeros and ones. A little bit I know about it. Hexokinase off. Protein off. Okay, so look at this. If this protein gene, right, is off, what's not going to get made in that cell? Telomerase is not going to get made. What's that cell going to lose every replication? Telomeres. That is the biological definition of aging, one of the key biological definitions. Now, you're doing great. What do cancer cells do all the time? So you get a cancer cell, right? What's it going to do? What's it going to do? What's it going to do? Well, would cancer be a problem if senescence was reached? Okay. Can cancer cells reach senescence? Absolutely. And thank the dear sweet Lord, some of them do, and they do not cause damage to us. What would allow cancer cells to continue to replicate? Pardon? Telomerase. So look, cancer is a mischievous set of mechanisms. In cancer cells, what did I talk to you about? Look at your P53 note for a second. What did you write down about P53 and all cancers in the human body? What's the mutations of all the different types of cancers in the human body? 256 different types. How many have a mutated P53? Did I talk to you about that? Yeah?
65%.
65%. Okay. That's a huge step in the process of what we call cancer. But over 90%, grade 12s, over 90% of all cancers in the human body have the telomerase gene that was turned off, turned on. So one of the things cancer cells do is activate the telomerase gene. What will that allow the cancer cells to do? You're going like this, Jacob. Replicate without reaching senescence. So here's a life lesson that we all learn in many different ways growing up. There's a lot of examples in life of this thing called the double-edged sword, right? Losing telomeres, okay, like in my body, grade 12s. Me losing telomeres. And slowly, more and more cells in my body reach senescence. They won't divide anymore. And I get older, I get slower. You go, well, that kind of sucks. You go, well, these are the cards we were dealt. But that mechanism protects us against cancer. So theoretically, let's just rewind the clock. Let's say every cell in our body always had the telomerase gene, always turned on. Okay, that's like immortality. Your cells just keep dividing, you never get older. Now clearly you want to get older than a day. So it doesn't matter, let's just have some fun. Theoretically you pick an age, right? Lauren, what age would you pick if you could stay one age?
35 maybe?
Yeah, most mid-30s. You still have your body pretty well functional. It hasn't started to fall apart yet. I agree with you. But if you had the body of a 30-year-old and your cells kept dividing, that would be great, but your probability of getting diseases would skyrocket. The more times your cells divide, what is the probability, even when P53 is functioning correctly, what's the probability of a mistake per cell division? 1 in a billion, right? Now, those are good odds, but if you're looking at it from well, you could keep living, each time that cell divides, there's a chance. The probability goes up. So, losing telomeres, the biologists try to tell us to make us feel good. Biologists say it's a break against disease. Cancer needs to fight that break by turning on a very special to make a very special protein to keep its telomeres long so it can continue to divide. So here's a question, write this down. If you're studying cancer, you're writing this question down, I'm studying cancer. How would you attack the cancer cell knowing what you now know about telomerase? So we're talking a fight here, going to Brady's theme of the day. So knowing what you know about telomerase, how would you attack cancer? What would you do theoretically? Any ideas? Yes.
Like you want to find a way to get it to senescence as fast as possible, and one way is burning up the telomerase gene.
Good. Think about what you know, attaboy. Think about what you know with hexokinase. So let's jot down his excellent insight there. Turn off the telomerase. Did you say gene or protein?
I see gene.
Okay, you could go either way. Let's deal with the protein because look what we have here. Hexokinase is a protein. You've seen a regulation mechanism. This patient has cancer. This protein is in their cell. Well, what would you try to do? Okay, good. You say with what? Turn it off. Good. With what?
Mr. Regan, can you please contact the main office? Mr. Regan, please contact the main office.
Okay, did I just hear mystery molecule?
Yeah.
Okay. So if we block telomerases, what? What do, okay, jot that down. Find a molecule that can bond to telomerase active site. Now you're getting somewhere. Okay, so would this be an example, grade 12s, of competitive or non-competitive inhibitors? That's right, so you find a molecule that could compete, right, for the active site. Or you would try to find a gene. Why? Do you have any understanding here of the efficiency? If you could get the gene turned off, how could that benefit the patient more than just turning off the enzyme? Yeah?
You turn off the enzyme, you have to keep turning off other as they're being produced. If you got the gene, it would stop producing.
Yeah, awesome. Did you hear that? You have to target the enzyme all the time. If you can get the gene turned off, all the cells that are, all the cancer cells that are produced in the future have that gene turned off. Right? So that is that. Okay, good. Any questions? Yes?
So, like, the fetus doesn't want, like, telomerase turned on, so, like, everyone else wants it off?
Yeah, so that's good. So in your body right now, my body, although you're still a teenager, from a biology point of view, you have an adult body per se. So you don't have, all the cells in your body aren't active with telomerase, but there are certain ones. I already mentioned one, so let's make a little list. What cells in my body have telomerase working right now as an adult? What was the one I mentioned? Sex cells, right? Do you know where your, I guess, I was going to say your immune cells. Okay, we'll pick that up tomorrow. Okay, thank you.